Hello everyone. So we are going to start with the three basic ideas, or you can say the process which generally takes place in the human body. So first of all, in this particular presentation we are going to deal with the catabolism of the heme. Moreover, we are going to deal with the pathological condition which is also known as the hyperbilirubinemia, and lastly regarding the jaundice. So let's start our presentation. So first of all, you see what is a heme. So as a definition, you see the degradation of the heme. It's the iron containing part of the hemoglobin, right? So now it is degraded into the bilirubin in our body by the reticuloendothelial cells, which are generally found in the microsomal fraction as well as in the cytoplasm sequentially, right? Now this bilirubin, it is the principal pigment of the bile. You should know that the bilirubin, it is the principal pigment of the bile, which is secreted by the liver, and it is excreted via the liver into the intestine. And the amount of the bilirubin production from different sources can be calculated or can be depicted as, first of all, you see the amount of the bilirubin that comes out from the destruction of the red blood cell. It is 250 mg per day. Moreover, from the infective erythropoiesis, around 30 mg of bilirubin production it takes place per day. Moreover, from other heme containing proteins, 30 mg also comes per day, and this is the total amount of the bilirubin which is secreted in the body. It is around 300 milligram per day, and 6 gram of hemoglobin per day it is destroyed. So 1 gram of hemoglobin is equivalent to 35 mg of bilirubin. So this is the overall calculation. So now you see catabolism of hemoglobin. So it takes place in the different steps. So let's discuss the steps one after another. So first of all, let's enlist what are the steps. So the first step is the destruction of the RBC to release the heme. It takes place with the help of the particular enzyme. This is known as a lysosome, right? Lysosome. The next one, it is the degradation of the heme into biliverdin. It is performed, or is this action, it is performed by the microsomal system. The next one, the next step, it is the reduction of the biliverdin into bilirubin. It takes place in the cytoplasm. Then the transportation of the bilirubin into the blood. Then hepatic uptake of the bilirubin, conjugation of the bilirubin in the liver, and finally the deconjugation of the bilirubin in the intestine. So these are the steps which we are going to discuss now in a sequential manner. So last, it is the excretion part. So let's start the destruction of the RBC. So this is also known as the extravascular hemolysis. So first of all, you should know that the physiological destruction of the old or aged RBC, it led to the phagocytosis of the RBC. Once the RBC has completed its life of 120 days, it will be phagocytosed. It will be taken up by the reticuloendothelial cells of the spleen. And from there, this hemoglobin, it gets degraded into two components, the heme and the globin, and this is done by the enzyme, lysosomal enzyme. So this heme, it is taken up by the OH hydroxyl system. Moreover, this globin, it is reutilized in the human body. The next one, in continuous or you can say in contused wound, the RBCs are generally taken up by the macrophages of the local tissue. We know that the macrophages, they act as a second line of defense in the human body. So once you have got any sort of wounds, the WBC will come, the macrophages will take up the RBC, right? Now what is the process there? So the next, it is the intravascular hemolysis, direct release of hemoglobin and heme in the circulation. Now what actually happens, the heme and the hemoglobin, they bind with the this HB, it is a hemoglobin, right? This hemoglobin and the heme, it binds with the hepatoglobulin and the and the hemopexin respectively, and it comes to the reticuloendothelial cells of the liver, bone marrow, and the spleen. So in this two way, the RBC, they are utilized for the production of the heme, or you can say the bilirubin. The next step, it is the degradation of the heme into biliverdin, and this steps, it takes up the help of an enzyme which is known as heme oxygenase, and which is found in the microsomal fraction of the macrophages. So this particular enzyme, it requires basically 4 to 5 molecules of NADPH. We'll discuss how this NADPH will work. and 3 molecules of molecular oxygen. Moreover, the process process it is the hydroxylation of the alpha methylene bridge between the 1 and the 2nd ring. The next step, it is the oxidation of the iron, right? The third step, it is the oxidation of the C of the methylene. C means the carbon of the methylene bridge into CO, and finally this particular bridge of the carbon, it breaks down. And then the next step, it is the release of the CO, and a ferric iron or a ferric iron later joins the iron pool in the body, and in this way the closed tetrapyral ring of the heme is opened into the open tetrapyral structure, and this biliverdin, the green bile pigment, major pigment in the bird, actually it is released in the cytoplasm of the reticuloendothelial cells. So now let's discuss whatever we have pointed out or jotted out in the previous slide. So this RBC, they are going to give the hemoglobin, right? Now from the hemoglobin, the heme part, it is first, this is the hemine, this is the ring. So this hemine ring here, see, 1 molecule of NADPH, it is utilized to convert it into heme. The next molecule, you see, we have already discussed the heme oxygenase. So here the next molecule of NADPH, it will be utilized with the presence of the oxygen to form the hydroxyhemine. Again, this oxygen will be utilized to oxidize the iron to finally get the biliverdin. So this is the steps which takes place. Again, if we see the steps, now the biliverdin which has formed in the previous step, now it will utilize one more NADPH molecule. We have already discussed regarding this carbon C, that the bridge it is going to break, right? So here you see NADPH will give the reduction or reducing equivalent, and finally the bridge has been broken, and it is leading to the formation of the bilirubin. Now what will happen to the bilirubin? You have seen here the bilirubin, right? Now the next step, you see this bilirubin, now it will get conjugated with the albumin, and this albumin, or you can say bilirubin albumin complex, will be formed. It is a carrier mediated process, and it will rich in the cell, right? This bilirubin Y and Z complex, actually it prevent the efflux, it prevent this component to going out or or escaping out from the cell, right? Now finally in the endoplasmic reticulum, this bilirubin, it is stored, or it will come. Now transport of bilirubin, we have already discussed. So alpha bilirubin is transported by the protein which is known as the most abundant protein in the body, which is known as the albumin, right? Now about 100 ml of the plasma can bind to 25 mg of the bilirubin. This is the approximate count. Now it binds, these albumins bind to the bilirubin tightly but non-covalently. You should know that the albumin will bind to the bilirubin tightly, but it is non-covalently. Moreover, the drugs like aspirin, sulfa drug, penicillin, etc., they always compete for the binding site. We have already discussed in the enzyme kinetics regarding the competitive binding of the substrate to the receptor or enzyme, right? So drugs like aspirin, sulfa drug, penicillin, they compete for the binding site, which can result in what? It will displace the bilirubin from the albumin. Now once this drug we have taken, this drug will displace the bilirubin from the albumin, leading to the toxicity. Now uptake of the bilirubin albumin complex by the liver. So the bilirubin is now taken up by the sinusoidal surface of the hepatocytes, right, by a carrier mediated process. We have already discussed about this thing in the previous slide, and the albumin is released back to the plasma because it is a carrier. It will be reutilized for the carrying of the hemoglobin, or sorry, the bilirubin. Now inside the hepatocyte cytoplasm, what will happen? Now this bilirubin, they binds with the ligand Y and Z, which we have already discussed in the previous slide. So this Y and Z, they belongs to the glutathione S transferase family, and that actually keep it solubilized and prevent the efflux efflux of it. So thus when this bilirubin is going to bind with the ligand, or you can say the ligandin Y and Z, then this will prevent its efflux. Now finally this bilirubin enters the endoplasmic reticulum and it prevents the efflux of it. The next step, it is the conjugation of bilirubin in the liver. So this process is carried out by, first of all, you see the enzymes. The first enzyme it is responsible, it is the uridine diphosphate glucuronyl transferase, which is also abbreviated as UDP. So UDP glucuronyl transferase enzyme UGT-A, or you can say UGT1A. Right? Now this enzyme it is found in the endoplasmic reticulum of the hepatocytes only, right? Mostly in the hepatocytes. This enzyme actually what it does, this UDP enzyme, it transfer the glucuronyl group of the UDP and UDP glucuronic acid to the COH group of propionic acid of the bilirubin, and this process it is called the conjugation of bilirubin. I repeat this process. This enzyme, this UGT1A, actually you see this enzyme it transfer the glucuronyl group of UDP glucuronic acid to the COH group of the propionic acid of the bilirubin, and this process it is a conjugation. We are going to get into details in the next slide regarding this thing. Now this bilirubin it is converted initially into monoconjugated bilirubin, which is also known as bilirubin monoglucuronide, and finally into the diconjugated form, that is the diconjugated bilirubin, which is also known as bilirubin diglucuronide, and render them water soluble. Thus it makes it water soluble. So this is the step what we have already discussed. So this is the bilirubin, right? This is the bilirubin. Now first UDP glucuronide transport will come, right? It will get added. Now this is the glucuronic acid. It will get converted into bilirubin diglucuronide. To 2 molecules of glucuronide it has get added to it, right? Now this UGT1 it is always, you should know that it is always located in the chromosome number 2. Moreover, the expression of the UGT1 in any individual it is reduced, it comes down or absent in some disease like Gilbert syndrome, Crigler-Najjar syndrome respectively. In such cases the amount of the bilirubin it is always high in the body, right? The activity of the UGT1 enzyme it is increased by some drug like phenobarbital, benzylpyrene and aminoquinones. So if you are on drugs like phenobarbital, benzylpyrene or aminoquinones, in such condition the production of the UGT enzyme it increases. Now it is useful in the treatment of the unconjugated hyperbilirubinemia. Thus you see the activity of UGT it is reduced by drug like pregnanediol, chloramphenicol, moreover sometimes the fatty acid. It also reduces the activity of UGT. Now the next step it is the secretion of the conjugated bilirubin in bile canaliculi. So what is actually happening in that process? See, this secretion it is helped by the ATP, or it is a ATP dependent transporter will act, that is MRP2. MRP2 transporter is nothing but the multi drug resistant like protein type 2 which is present over the membrane of the bile canaliculi, right? In Durbin-Johnson syndrome MRP2 is absent in this particular disease. Normally you should know that very trace amount of the conjugated bilirubin escape from the liver to the systemic circulation, because bilirubin it is always remain as intact. We have already discussed, as the albumin it will get deconjugated from the bilirubin once it will reach the liver, because this albumin will be reutilized again for the transport of the bilirubin, right? Now in the bile diconjugated bilirubin, the conservation or you can say the bile diconjugated bilirubin, the concentration of the diconjugated bilirubin it is always high as compared to that of the monoconjugated bilirubin in the human body. Moreover, the unconjugated bilirubin if it is present, then you have to go through the phototherapy. Now deconjugation of the bilirubin in the intestine. Now this bilirubin will be deconjugated. So this bilirubin diglucuronide, now it will be deconjugated, it will be, the glucuronide molecule will be removed from it. So what enzyme it is going to act? Here basically the beta glucuronidase of the bacterial flora terminal which is present in the ileum and the colon, they are going to deconjugate this, means the bilirubin will be produced as bilirubin, this like glucuronide molecule will be removed, right? The deconjugated, it led to the production of the deconjugated bilirubin and glucuronic acid. and this reduction is done by the bacterial flora again to convert it into urobilinogen, or you can say stercobilinogen. So basically you see deconjugated bilirubin, if it get oxidized, it forms the biliverdin, right? It gives the green color of the stool. Similarly, if the reduction of the bilirubin takes place by the, we have already discussed the reduction, it takes place by the bacterial enzyme, right? Now if it don't takes place, then the color of the stool will become green. But if the reduction takes place, it will form the urobilinogen, or you could say stercobilinogen, which is utilized in the stool. The color of the stool, it comes from this. So finally it will form the stercobilinogen, or you can say stercobilin. So similarly, and if it don't get utilized, then this can go to the liver. Again, the liver will utilize it in the CBD with the help of the enzyme EHC, which again comes to the stool. And similarly, see, the color of the stool, it is coming yellow due to this stercobilin. But what about the color of the urine? So the liver actually, if it synthesize, or if the liver get a chance to synthesize, means oxidize it, so then this liver, it is going to send it the bilirubin in the systemic circulation, right? From there it will go into the urine as a urobilin. The next condition, which is also known as the hyperbilirubinemia, so it is a pathological condition. So let's discuss what is this. So hyperbilirubinemia, it is nothing but it is an elevated level of the pigment of the bilirubin in the blood, right? So it occurs when the serum bilirubin level, it crosses 2 mg per deciliter of the blood. Now what are the condition or consequences of this? If it happens, then yellowing of the skin will take place, yellowing of the sclera, means white of the eye, which will take place, and the mucous membrane, even in case of the jaundice. So a sufficient elevation of the bilirubin produces jaundice, we know that, right? Now this hyperbilirubinemia, it is a biochemical finding, but jaundice, hyperbilirubinemia, I again repeat, the hyperbilirubinemia, it is a biological finding. Means you can check the level of the bilirubin and you can come to the conclusion for that it is the person is suffering from hyperbilirubinemia. But jaundice, it is a clinical finding, means the jaundice, once the jaundice will develop, you will find clinical symptoms for increase in the bilirubins in the blood, right? Now bilirubin metabolism, how the metabolism of the bilirubin it takes place. So first of all, hepatocellular bilirubin transport. So how it takes place, let's discuss step by step. So this part we have already discussed regarding the conversion of the heme to the bilirubin, right? Now if we see this structure here, you see here how it takes place. The albumin bound bilirubin, here albumin bound bilirubin, you just see, you will start from here, you will end up here, right? Now albumin bound bilirubin in the sinusoidal blood, it passes through the endothelial cell fenestrae. It will pass through the endothelial cell fenestrae to reach the hepatocyte surface. Now entering the cell by both facilitated, it will enter the cell by both facilitated and simple diffusion process. Now within the cell, it is bound to the glutathione S transferase and conjugated by bilirubin UDP glucuronyl transferase UGT1A1 to mono and diglucuronides. This is the UGT1A1, right? This is the conversion. So which actively transported across the canalicular membrane into the bile. So here you can see here I have given the abbreviation. ALB stands for the albumin, then the BDG stands for the bilirubin diglucuronide, BMJ it is the bilirubin monoglucuronide. Here the abbreviation are given here. Now you see the transport of the, or you can say the transfer of the bilirubin from the blood to the bile, it involves 4 distinct interrelated steps. So what are the interrelated steps? First step, it is the hepatocellular uptake, right? The next step, it is the intracellular binding. The next, it is the conjugation, and finally the biliary excretion. So these are the 4 steps for the metabolism of the bilirubin. So let's start with the bilirubin and its breakdown. So we have come to know that this from the heme you are going to get this bilirubin. Now this bilirubin, it may get converted into bilirubin diglucuronide by conjugation of 2 glucuronic acid molecule, right? And even similarly the bilirubin which is present in the bile, right? Now this bilirubin, it get converted into urobilinogen. From here it is transported to the kidney as a urobilinogen. From here it is going out as a urobilin or a stercobilin, right? Now you see enterohepatic circulation. What is that? It's a very important part. We are going step by step. So first let's start with the step 1. We have seen, you can see here I have given you as a 1, 2, 3, 4, 5 like this. The first step, it is the senescent of the red cells are the major sources of the heme protein. We know that, right? Now the breakdown of the heme to the bilirubin, the step 2, occurs in the macrophages of the reticuloendothelial cells, right? And once the breakdown will takes place, the unconjugated bilirubin, it is transported through the blood to the liver, right? The fourth step, it is the bilirubin is now taken up via facilitated diffusion by the liver and conjugated with the glucuronic acid. Till this step we have already studied. The fifth step, it is the conjugation of the bilirubin is actively secreted, or you can say conjugated bilirubin is actively secreted into the bile and then to the intestine. Now it is going to the intestine, right? In the intestine, what will happen? The glucuronic acid is removed by the bacteria. Regarding this thing, we have already studied. Now the resulting bilirubin is converted into urobilinogen. This is the sixth step. The seventh step is nothing but some of this urobilinogen is reabsorbed from the gut and enters the portal blood. Some of this urobilinogen, it is reabsorbed. You should remember that. The eighth step, it is that a portion of this urobilinogen, it participates in the enterohepatic urobilinogen cycle, right? It will be again sent back to the liver. Moreover, the remainder amount of the urobilinogen, it is now transported by the blood to the kidney, where it get converted into yellow urobilin pigment, and it is excreted, giving the urine its characteristic color. You see here, it is urobilinogen, it is coming out in the urine, giving the characteristic color of the urine, that is a pale yellow. Moreover, the last step, it is the urobilinogen, it is oxidized by the intestinal bacteria to a brown pigment which is known as stercobilin, and it comes out from the body in the form along with the fecal matter. Thus the stool, the color of the stool, it is due to the stercobilin. Now, hepatocellular conditions that may produce jaundice. So what are the conditions? Let's discuss. First of all, the jaundice can be produced by, or you can say viral hepatitis. It can be produced by the virus. What are the types of the viral hepatitis? Hepatitis type A, B, C, D, and E. It is generally caused by the virus like Epstein-Barr virus, cytomegalovirus, herpes simplex, right? Moreover, alcohol that can induce the jaundice-like symptom in the human being. Next one, it is the drug toxicity. So what are the drugs? So predictable drug toxicity, it is dose dependent. In case of the acetaminophen, it is nothing but it's the paracetamol, right? It is hepatotoxic in nature, we know that. But this is a predictable. We know that this paracetamol has a tendency to induce or affect the liver, right? But unpredictable or idiosyncratic drug toxicity is caused by drugs like isoniazid, right? The next one, it is the environmental toxins. So there are different environmental toxins which we used to take in our life, means knowingly or unknowingly. The drug like, or you can say component like vinyl chloride. Sometimes some people used to take the Jamaica basti, which actually have the pyroxolidine alkaloids. That also can cause the jaundice. Kava, it's a herb. Kava, it's just look like a betel leaf. So it is generally used by the tribal people for the treatment of the postmenopausal syndrome. The next one, it is the wild mushroom. Example, Amansia phalloides. So they may also induce the jaundice. The next one, it is the Wilson's disease and autoimmune hepatitis. So in this condition, the jaundice can get induced in a normal human being, right? Now the cholestatic conditions that may produce jaundice. So what is cholestatic? We all know that the decrease in the bile flow. It is nothing but the decrease in the bile flow. So if the bile flow it is less from the liver, then it can also produce a jaundice-like conditions. So it has been divided. This cholestatic condition has been divided into two parts. The first part, it is the intrahepatic. So intrahepatic condition, it induces the viral hepatitis. So this can led due to the fibrosis, or you can say fibrosing of the cholestatic hepatitis. It is generally happens in hepatitis B and hepatitis C type. Next, the hepatitis A, Epstein Barr virus or cytomegalovirus. They also cause the viral hepatitis, and that can lead to the cholestatic, and which finally lead to the jaundice. The next one, it is the alcoholic hepatitis. We have already discussed drug-induced toxicity. So pure cholestasis, it is generally induced by the component which are anabolic or contraceptive steroids. They can cause the cholestatic hepatitis, right? Next, the cholestatic hepatitis sometimes also induced by the drug like chlorpromazine, erythromycin, estolate, right? The next one, it is the chronic cholestasis. This chlorpromazine and prochlorpromazine, they generally cause the chronic cholestasis. So what actually happens there? The primary biliary cirrhosis, it takes place with the drug like chlorpromazine and prochlorpromazine if you have given to any person. Moreover, primary sclerosis cholangitis. So what is primary sclerosis cholangitis? It is a complication of the ulcerative colitis, in which actually what happens, the bile duct develop irregularities and narrowing, leading to the less amount of the bile flow out of the liver. The next one, it is the vanishing bile duct syndrome. So in these are the symptoms which are generally found in the chronic cholestasis, and this can lead to the jaundice. The next one, it is the chronic rejection of the liver transplant. If the liver transplant, it takes, means if the body is rejecting the liver transplant, that can also induce the jaundice. The next condition, it is the sarcoidosis. So what is sarcoidosis? So it is generally, you can say, it is a chronic disease of unknown cause, characterized by the enlargement of the lymph nodes in many parts of the human body, and the widespread appearance of the granulomas derived from the reticuloendothelial system. This is a pathological condition. This is the sarcoidosis, right? Then again, drugs. By the drugs, it can also cause sometimes inherited conditions, like it can also come from the parents also, like the progressive familial intrahepatic cholestasis. If it is there in the family history, then the person has a chance of developing cholestatic jaundice. The next one, it is a benign or recurrent cholestasis. The next one, it is the cholestasis during the time of the pregnancy. Now again, in the intrahepatic conditions, we have different conditions like total parental nutrition. It also induce non-hepatobiliary sepsis, benign post-operative cholestasis, then the paraneoplastic syndrome, then veno-occlusive disease, graft versus host disease. This all may induce the cholestatic, and which actually lead to the production of, or you can say, development of the jaundice. The infiltrative disease, they also develop the condition of the jaundice. For example, tuberculosis, lymphoma, or amyloid disease. Apart from that, infections like malaria and leptospirosis, they can also induce the jaundice in a normal human being. The next condition, it is the earlier one, that is the intrahepatic. The next one, it is the extrahepatic. So extrahepatic condition which induces the jaundice includes the cholangiocarcinoma. All these are actually all are the cancers. The cholangiocarcinoma, pancreatic cancer, gallbladder cancer, ampullary cancer, even malignant involvement of the porta hepatis lymph nodes. These all may induce the jaundice. Apart from that, these are the malignant condition. Actually, malignant condition, I hope you people know what is a malignant condition. It is the controlled growth of the cancer. It generally takes place in the later stage of the cancer. And regarding the benign, so benign conditions include the cholelithiasis, post-operative biliary structures, primary sclerosis, cholangitis, then the chronic pancreatitis, AIDS, cholangiopathy, Mersey syndrome, parasitic disease, which includes the sclerosis. So these all conditions, these are the benign. These can also induce the jaundice, and these are the malignant conditions. So finally, thank you everyone. We have completed the 3 topics togetherly. I hope you all like my video. If you like the video, press the like icon. You can share this video with your classmates and colleagues, and for further notification from this channel, you can press the bell icon. Thank you once again.